Hello, everybody. Let's go ahead and get started. Hi, my name is Tom Ulrich. I'm the Associate Director for Science Communications here at the Broad, and it's my honor to welcome all of you here tonight and everybody who is watching on YouTube to the third of our Broad at 15 talk series. Tonight, we are joined by Ben Neale and Beth Stevens. Both Ben and Beth are Institute members in the Stanley Center for Psychiatric Research here at the Broad. Ben is also a geneticist with the Analytical and Translational Genetics Unit at Mass General Hospital, and Beth is a neurobiologist at Boston Children's Hospital. Together, they're going to talk about the evolving understanding of psychiatric disorders, how genetics is guiding us to new biological insights, and the challenges of studying the brain. If you have any questions for Beth and Ben after their talk, we ask that you raise your hand for the mic runners. You can also text your questions to the number that's going to be on the screen. And if you're watching on YouTube, you can also send in your questions by YouTube chat. And if you're tweeting, I always forget to ask this one. If you're tweeting, we do ask you to use the hashtag BroadAt15. Without further ado, coming up, Ben.
Thank you, Tom, and thank you all for joining me tonight. I'm going to talk to you about what genetics is teaching us about mental illness. But to start, I think it's important to start with really what is mental illness. When we think about mental illness, you know, there's this sort of dry definition of things where you're having a problem with your thought or mood or how you interact with people. But actually, in many ways, mental illness is the most personal of illnesses when we think about people getting sick. It disrupts not only ourselves, but our families' lives. And I'd actually like to take a moment to kind of engage with the audience a bit and raise your hand if you or a loved one is affected by mental illness. Yep. So just look around. That's almost everyone in the auditorium. And these are really, really common. You know, these are amongst our most common disorders, our most common illnesses. They represent one of the single largest sources of disability-adjusted life years. That's the sort of health economist perspective on how much of an impact is a disease or disorder having on you. They represent that biggest source of disability-adjusted life years in the Western world and are one of the main early causes of mortality and morbidity, particularly in the young, with suicide ranking amongst the top causes of death for people between the ages of 18 and 35. Mental illness is really hard to study, and it's hard to study in a lot of ways. And we've actually sort of come a long way in that study, and I'm going to try and take you through some of that journey tonight about how we're trying to learn a little bit more about mental illness, how we think about it, and how we can start to lay the groundwork for improving the ways that we characterize it, we diagnose it, we classify it, and hopefully, eventually, the way that we treat it. So as a kind of origin story for diagnoses, you know, like superhero films are all the rage right now, so origin stories are sort of common territory. The origin story for mental illness traces its lineage back to this guy, Emil Kraepelin, who is active in sort of Central Europe in the, you know, kind of 19th, early 20th century. And prior to Kraepelin and many others at this kind of same time, there was a recognition that people would suffer from mental illness and they would have this form of madness. And they would just call it madness and they would be, you know, dealt with in a sanatorium sort of setting, and that's about the extent of the way that we were thinking about this group of people that touches everyone in our lives, as we saw a little bit earlier. And Kraepelin said, well, we should maybe try and move beyond just this idea, this concept of madness, and break it down into a little bit more of a structure, impose some kind of structure on these illnesses. And he created this so-called Kraepelinian dichotomy between manic-depressive illness, which at the time included what we call bipolar disorder, as well as major depressive disorder. And then on the other side had dementia praecox, or schizophrenia. Now, for the rest of the talk, I'm primarily going to focus on schizophrenia. And that's not because all of the other mental illnesses aren't really important. They really are. But I think it'll help illustrate some of the approaches that we're taking to try and understand how these diseases come to be, how people get sick and affected by them more generally. And when Kraepelin was creating this dichotomy between manic depressive illness and dementia praecox, one of the primary arguments that he used to justify this delineation was that there was a tendency for those two different kinds of disorders to run in families. So that is to say that patients that had dementia praecox tended to have more other relatives that were affected with dementia praecox. And the same held true for manic depressive illness. And so that idea of things running in families was even recognized back then, over a century ago, and was a kind of core part of the belief that there is a biological basis, at least in part, for these disorders. Okay, so biology plays some role, and we just don't necessarily understand what it is. So we can take that kind of idea of twin in families, of running in families as a way of getting some indication that there might be some genetic influences, and try and formalize it a little bit. So on the left is a picture of a guy named Francis Galton, who really, termed regression. So when you run a regression, that's, Galton is the person that named it as such. And he was a Victorian gentleman scientist that spent a lot of time thinking about familial similarity and used the observation that parents that were tall tended to have tall kids and parents that were short tended to have short kids, but not quite as tall or quite as short as their parents were, and used that as an argument in favor of this idea that genetic variation, even though the structure of the genome wasn't known, even though the concept of DNA didn't really exist, the extent of our understanding of genetics was really down to Mendel and his peas. But it was moving away from those discrete characteristics, like whether you're a yellow pea or a green pea, into continuous traits, things like height and BMI, thinking about those kind of anthropometric traits. Those continuous traits also run in families, but there's no one variant that makes me 6'4". That's not the way kind of continuous traits like height work. Rather, it's this idea that there are lots of genetic effects that are scattered out there in the genome that they didn't even appreciate, but they knew in a sense, inductively, that there were lots of these genetic effects and that the similarity in family members can be one way of indexing how much genetic influences various disorders or continuous traits, things like height. And what you see on the right side of the slide is the relative rate, the overall lifetime risk of developing schizophrenia, conditional on having a relative with schizophrenia. So if you're a monozygotic twin or an identical twin, you've got about a 50-50 shot of developing schizophrenia if your co-twin has schizophrenia. And you can contrast that with a sort of random person from the general population where there's about a 1% shot. So that's a really, really big difference in the chances of schizophrenia, conditional on whether or not your relatives have schizophrenia or don't. And that, again, is this idea that genetics is playing some kind of a role, that there is a genetic component to the risk for developing schizophrenia in the population. The other thing that plot shows on the right is that the risk falls off as your sort of familial similarity falls off. So you're more genetically similar to your siblings than you are to your aunts and uncles or cousins, than you are to your second cousins and third cousins. And you can see that as you move from first-degree relatives to second-degree relatives to third-degree relatives, you see a lower increase of the risk. So the further away someone is in your family tree that has schizophrenia, the comparatively less likely you are to have schizophrenia yourself. And that is also consistent with this idea that there are lots of small genetic effects that are influencing schizophrenia more broadly in the population. Now we have to move from this idea of families into actual genetic variation. And that's where the last, 20 years of progress in human biology, and really the 15 years that the Broad has been playing a major leading role in these activities, has really come to pass. We've seen the sequencing of the human genome at the turn of the 21st century. Then following that, there were efforts to characterize what the common genetic variation is in the population through projects like the International HapMap project that more or less laid out what variants are common, tried to capture as many common genetic variants as possible, and understand the correlation between those genetic variants so that we could design genotyping arrays to efficiently capture all common genetic variation in the population for maybe something like $50, which is about what you'd pay for now. And then on to next generation sequencing. And with sequencing, we have this transition from looking just at common genetic variation, like we do with our GWAS arrays, those pictures in the bottom middle of the slide, to sequencing, where we can capture almost all genetic variation simultaneously in an individual, going all the way down the allele frequency spectrum to the rarest variation. Now, this is the technology that lives underneath collecting genetic information in the population. But what do we do with it? Well, the scale of the challenge is massive. We've got 3 billion bases. We've got 18 to 20,000 genes. There are maybe 20 million common variants. It's a big search space. But the way that we approach that is to systematically ask the question whether a genetic variant is more common in people with a disease or disorder versus people that do not have the disease or the disorder. So very simply, we ask for each genetic variant, is it more common in people with schizophrenia or not? And that's the same question that we ask systematically at each one of those 10 or 20 million common variant sites that we're looking at across the genome. And the first, or one of the first such efforts to look at the genome-wide association data for schizophrenia was published in 2009 by the International Schizophrenia Consortium. And that looked at 2,600 cases and 3,300 controls. And you're going to see a sequence of these kinds of pictures where what we've got is this sort of Manhattan plot here. And a Manhattan plot ideally is meant to look like Manhattan. So you ideally would see skyscrapers in Manhattan. Things look pretty flat here. And then you have this line, this red line here. And what that red line represents is the sort of level of evidence that we find sufficient to believe that that SNP, or at least SNPs in that region, are actually truly associated to schizophrenia. And in 2009, what was at the time one of the largest single experiments in psychiatry as it relates to genetic evaluation sort of came up a little flat, right? This doesn't look like, Manhattan. Maybe it looks like Manhattan, Kansas, rather than, Manhattan, New York. And we don't see anything really breaking through this barrier to be compelling evidence that it's actually associated to risk for schizophrenia. And the big message here is written down here at the bottom, is that we're going to have to work together. And I think that's another element that the Broad in particular has really tried to encourage and foster as much as possible, is this idea that if we as a scientific community can work together, we can make these discoveries faster. And by making these discoveries faster, we might get to a better understanding of why people get sick. And that will then in turn lead to, hopefully, our ability to understand ways to diagnose and treat patients. So we got to work together. And so how do we work together? Well, as a geneticist, we spend a lot of our time in these large-scale consortium activities where we're linking together with hundreds of other scientists scattered across the globe, each of which has either collected some schizophrenia patients or some controls and shared that data with us to try and advance or accelerate our understanding of the genetic basis. And the PGC is really one of the sort of primary pioneers of this kind of approach. And it cuts across a huge number of different disease or disorder areas, ranging, as you see over here on the left. And the focus on it analytically is to try and extract as much important information and insight from these data sets as possible, including analyses of common genetic variation or looking at copy number variation, or even probing what the nature of the relationship is between different kinds of mental illnesses at a genetic level. We can do those sorts of things as well. And this effort to work together has actually started to gain traction. We've moved from having 0 genome-wide significant loci, 0 things that we were absolutely convinced or compellingly convinced of their strength of evidence as being relevant to schizophrenia, now here to 108 published in Nature in 2014, up to now continued progress to date, looking at over 65,000 cases and identifying more than 240 loci across the genome. Now, this looks a lot more like Manhattan, New York, right? We got all of these skyscrapers. Each one of these skyscrapers represents a genetic variant somewhere in there that is influencing risk for developing schizophrenia. We don't, in most cases, understand how or why or what, and that's actually probably the biggest challenge of the next 15 years, is how do we go from these kinds of results and insights into a clear understanding of the biological basis, not just of schizophrenia, but also of depression, of autism, of ADHD, of bipolar disorder, or more broadly, things like diabetes or inflammatory bowel disease, that the full range of human health can be subject to this kind of interrogation, and we can learn things about why people get sick by employing these kinds of technologies. Now that's the common genetic variant side of the story, but there's that other technology that I raised at the outset of sequencing and looking at rare variation. Now when we look at common variants, we mostly look at, we try and look at all common variants all at once, and when we do that, most of the common variants lie outside of genes. Actually, most of your genome is not inside of a gene, it's outside of a gene. This historically has been called junk DNA. That is a terrible name. It is a misnomer. We do not like to think of it as junk DNA. It is clearly where most of the action that is driving risk for schizophrenia lives. And so really definitely should not be considered junk because it's clearly important. But nevertheless, genes themselves are extremely powerful tools for understanding. We know a lot more about genes than we do about the non-coding genome. And with that in mind, we then also adopted this same approach of looking at variants that are more common in cases versus controls. But here we'll turn our attention to specific gene variants that are derived from sequencing, so we can look at extremely rare mutations in the population. And that's an important class that we didn't really have the ability to interrogate earlier on when we were looking at these common genetic variants. So this is the schema consortium. There's a picture of TJ, who's the postdoc working with myself and Mark Daly, who's really been driving forward these analyses. And the sort of gene-wise plots and analyses of rare coding variation is also starting to deliver genes. So that exactly the same approach that we used for common genetic variants is working for rare coding variants. But rather than giving us a whole bunch of puzzles about what the gene really is in the region, it is pointing very, very specifically to individual genes based on coding variants in those genes. And so here we've got a Manhattan plot. Now each one of these dots represents a gene rather than a common genetic variant. And what we see is that there are clear hits that are emerging. This sort of similar line here of truth versus not truth, which is a little bit of an oversimplification, but basically that's the strength of evidence that we feel is necessary to be confident in its involvement in risk for schizophrenia. We see 10 individual genes that are popping up above that line, and these are really important discoveries. They are teaching us things about the risk for schizophrenia that we did not know before doing this work. And so we can kind of compress 15 years worth of schizophrenia genetics onto this single slide. And this slide's got a lot of information. I'm going to try and walk you through it. So on the x-axis here, we've got the minor allele frequency. So that's how common this variant is in the population. And it ranges from extremely rare, 1 in 10,000, sort of rare, maybe even rarer than that, up to the much more common, or 50%. So a 50% allele, basically, a quarter of the population will have one version, homozygous, so 2 copies of one version. Because we're diploid, we get stuff from mom and dad. Half the people will have 1 copy of one allele and then the copy of the other allele, so they'll be heterozygous. And then another quarter of the population will be homozygous for the other one. And so all of these little blue dots that you see over here, those are the common variants in that Manhattan plot, but now shown based on what their frequency in the population is, and then on the y-axis, what the odds ratio is, or what the effect size, what your increased chance of having schizophrenia would be if you happen to carry a copy of this variant. Now these are really, really common, and that means that everyone in this room has schizophrenia risk alleles. We all have schizophrenia risk alleles. That is part of our life, that is part of our species, that is part of our population, and we should not lose sight of that. As we drift further down the allele frequency spectrum, it suddenly becomes possible that maybe these variants don't exist in this room, but they certainly exist in the population in general. And you can also see another sort of pattern emerging. So if we look at the odds ratio here on the y-axis, as we go from common to rare, we see that the effect sizes go up. And there are a lot of reasons for that, but kind of one of the primary reasons is that we only have the ability to detect rare variants that have really big effect sizes. So there are rare variants that will have effect sizes down here. We just don't have enough people to look at them. And then there are things about whether or not you're going to have children based on if you have schizophrenia. And if you look at population level estimates of fertility and fecundity, you see the clear impact there because of its impact on people's lives. And that's really meaningful as well. So here's like 15 years of work compressed into a single slide, but what are we learning from it? So the emerging biological insights that are really starting to come out of this work that I think poise us for another very important 15-year journey is that the synapse is really, really important from a genetics point of view. Now, synapses are part of neurons. They're actually the way that neurons talk to one another. So here's a picture zoomed in on a synapse. And when I mean talk to one another, neurons talk to each other in a couple of different ways. Actually, probably more than a couple, but let's just put it into two categories. One is through electrical signaling, so they flow ions. That's one way that they talk. And then the other way is through chemical signaling. So things like neurotransmitters and the like are also used to communicate between neurons. And the synapse is really where the action for that kind of communication is happening. So here's a little zoomed in picture of maybe a calcium channel, ion channel there in the synapse. And from the common variants, we're seeing strong evidence of a bunch of synaptic genes. And from the rare variants, we're also seeing strong evidence of synaptic genes. And so to take us that step further, to explore the synapse and learn about some of the core basic concepts of neurobiology, I'm going to turn it over to Beth Stevens, who's going to pick up for the second half of the talk. Thank you, Beth.
Okay. Great, thanks. Okay, great. So I'm a developmental neurobiologist and actually spent most of my career studying this question of how synapses are functioning and how they develop and how the brain is wiring up. And I think some of that insight is starting to come together with some of the genetics you just heard about today. And I'm going to tell you one such story. But I want to sort of step back and kind of dig into this a little bit deeper. And the fact is that we all appreciate, right, that the connections between neurons, synapses are the basis of communication in the brain. But if you were to zoom in on a synapse, you'll see that there's actually a lot of molecules there, a lot of machinery there that makes the connections or the junction between neurons work. And any one of these channels or molecules, if they're disrupted, can affect synapses and their function, which could have obviously large and long-term consequences. And it's actually been shown, interestingly, that synaptic loss and dysfunction is a hallmark of many neurological disorders, not just for neuropsychiatric disorders and neurodevelopmental disorders, but even more broadly. And so it's actually, in the case of neurodegenerative disease, for example, it's the strongest correlate of cognitive decline, this idea that you lose synaptic connections or synaptic function even early on in the course of disease. So there's a lot of evidence for this, both in human studies and in animal model studies. But the question of what makes synapses vulnerable is still an open question in all of these diseases. And so understanding how synapses work and understanding the biology underlying the emerging genetics is really one of the major goals of the work that's going on here at the Stanley Center at the Broad, but also one of these challenging questions. And why is it challenging? So it seems pretty straightforward, right? Like you've got some of these genes. In some cases, the genes have been identified. They map to a particular channel or a certain receptor. And then you're like, great, you can go in there and start to understand how it works. Well, it's not that simple, in part because the brain is incredibly complex, right? The brain is made up of 100 billion neurons, 100 billion, trillions of synaptic connections. So every neuron makes thousands and thousands of connections. And somehow, each neuron forms synapses, the right synapses at the right time, that somehow lead to circuits. And those circuits actually regulate specific behaviors or cognitive functions in ways that we're still beginning to try to understand, right? So, of course, trying to understand which synapses or which circuits are vulnerable is one of the major questions in the context of neuropsychiatric disease. And so, clearly, the complex circuitry makes it challenging. But the other thing that makes it even more challenging than in other disease areas, such as cancer, for example, is the inaccessibility of the human brain. And the fact is, you can't just go in there and pull out a piece of brain and biopsy it and ask what's wrong, nor can you, unfortunately, do we have the tools to be able to put someone into an imaging scanner and see synapses, or be able to look at these molecules over time. And so that has limited our ability to know exactly what's happening. Moreover, and this is actually a really big one that I'll come to later in my talk, is that we lack credible disease models. Because we can't access the human brain, because we can't actually, and the human brain is clearly more complex than many other organisms, the fact that we don't have very credible models makes it very challenging, especially in the context of what you just heard from Ben, in that this is a complex polygenic disorders. It's hard to model that. We'll get back to that in a moment. And I think equally important, and because we lack the biological insight right now, we don't have biomarkers and ways to be able to know when and who's at risk, and be able to predict who's going to have neuropsychiatric disorders. So that's another big challenge. So the big question is, how do we map these genetic variants that you just heard about today to specific functions? Right? So somehow we have to figure out, and in some cases we don't even know what the gene is. So we need to understand what the gene is. We need to know what that gene does and how it relates to risk, how it's functioning in a cell. We need to therefore understand biological pathways. That's going to lead us closer to understanding mechanism if we understand how it fits into a pathway that controls certain functions. We need to know which cells, right? I just told you there's 100 billion cells. Which of those cells might be more vulnerable? Could there be certain types of neurons or glial cells that are more vulnerable than others? Could there be certain brain regions or synapses or circuits that are more vulnerable? And then ultimately, we need to understand when things go awry, right? So the timing is important. And since we don't have ways to track and know, it's not clear yet when this happens, when the brain starts to become dysfunctional. However, there's quite a lot of evidence in the context of neurodevelopmental and neuropsychiatric disorders that a lot of the problems arise during development. These are developmental disorders. And so this is really what I want to talk to you about in the next few moments, is just trying to step back and just ask, what do we know about brain development? What do we know about the development of the brains of individuals that do have some of these psychiatric disorders? So one thing we know is that the brain develops over many years of life, right? And if one were to go in, and this has been done mostly through you looking in postmortem analysis of brain and be able to look at the changes in circuitry and synapses over development, it's clear that we start off with a rather simple architecture of synaptic connections, as shown in this first image here. But then over the course of development, there's a lot of formation and building the brain, building these connections, right? This is synapse development, synaptogenesis, they call it. And then after a certain time in development, some of these connections get pruned away or refined away. And so there are different critical periods in the brain in which this remodeling is happening. And this remodeling is actually really important. You may have heard about the term plasticity, right? We all want our brains to be more plastic, especially as you get older. You want to be able to, for example, learn new language. My daughter, who's 8, 9 years old, fluent French. She's been doing that since she was in first grade. Now, if I try to learn French, it's a disaster. Why is that? It's because I've passed my critical period. It's not that I can't do it, I just can't do it as well, right? And so while this plasticity can be a very good thing for learning new things, one idea is that maybe this plasticity is also what's giving rise to vulnerability. Because while all these changes are happening in the brain, this also sets up, if there's genetic risk, in combination with other factors, at the right time, this could lead to defects in brain development. And it's been shown in humans, for example, through brain imaging studies, and a series of studies that actually tracked, using functional MRI and other brain imaging, children over the course of development. Every 2 years, they looked at their brains and asked how they change. And what's clear is different parts of the brain develop at different times. So areas like the visual system and sensory systems tend to develop and mature pretty early in life, but quite interestingly, areas involved in executive function, right? This is in higher cognitive functions, like the frontal cortex, as shown here. It turns out that those brain regions continue to develop and mature into early adolescence, into early adulthood even. So this is the last part of the brain, these important circuits that are involved in higher cognitive function, cognitive flexibility, working memory, things that we know can be defective and go awry in disorders like schizophrenia and neuropsychiatric disease. It turns out that these brain regions are the last to develop, raising the question of whether this could also relate to the vulnerability. And indeed, there's been some other evidence, although not a lot, but some evidence that has shown and demonstrated that the gray matter in your cortex is where all the synapses are packed in. If you look, for example, at this image of twins, one that has schizophrenia and one that does not, you can see, hopefully, that there's a difference in the gray matter. There's more thinning. There's more spaces there in that individual with schizophrenia. Again, suggesting the possibility that there's too much thinning or synaptic loss going on. And quite intriguingly and importantly, there's evidence that this is already apparent at the first psychotic episode. So this may be an early sign that synaptic defects are happening. And indeed, based on the work I just told you about, many different pieces of evidence, these are a lot of indirect strands, Erwin Feinberg, over 30 years ago, proposed the pruning hypothesis for schizophrenia based on the work I just told you about today, and the fact that he noted that these changes in synaptic pruning and synapse loss were correlating with this period of adolescence, which is, again, when the onset of these disorders actually happens. So what's explaining this particular time in development? Now, there's been a couple of other pieces of evidence, again, indirect, that have also observed in the frontal cortex, those same brain regions I just told you about that are involved in executive function and cognitive flexibility, there's been evidence that at least some individuals with schizophrenia have a sparsity or a decrease in these bumps, these spines, which are synapses. But again, this is indirect. The question of whether the synaptic loss is cause or consequence for many years has still been unresolved and is still unresolved. And that's in part because by the time we get the brains of these individuals, it's many years after the symptoms have emerged. And we haven't had any way of asking when this happens and how this happens. And this is where the human genetic results that I'm about to tell you about are starting to provide new insight. Many of these genetic variants, in particular those that are involved in synapse function or synapse development, could perhaps start to put these two pieces together. And in particular, as Ben mentioned, the strongest genetic association, the tallest peak on the Manhattan plot, is in the MHC locus on chromosome 6, right here. And if you were to zoom in on that locus and ask what genes are in that locus, there's actually hundreds of genes there, shown here on the right, on chromosome 6. And quite interestingly, many of the genes in that locus encode for different immune-related genes, genes that are traditionally thought of to be regulating the innate immune system or the adaptive immune system, which raise a number of intriguing hypotheses or ideas about the cause of schizophrenia. But the problem is there's been really no traction on identifying which of those genes is actually underlying the genetic signal and the risk. And so it didn't follow any mathematical or statistical pattern explained by known variants. So what could underlie this signal? And this is really where exciting and pioneering work by Stephen McCurl and his graduate student Ashwin Sekar and others here at the Broad Institute, they actually mapped one gene in that locus as explaining a lot of that strong signal in the MHC locus. So again, now what I'm showing you is zooming in on the MHC locus. Here, you can see there's one area where there was strong genetic signals right near this gene called C4, which is a complement gene, which you're probably thinking, what is that, right? This is an immune-related gene, traditionally involved in innate immunity. And it turns out there's a number of interesting reasons why Ashwin and Steve honed in on that gene. And the one reason was that this gene was actually quite different than a lot of other genes in that there's a lot of different genetic forms. An individual, any of you, could have C4a, there's two genes, C4a and C4b. And depending upon the structural combination or haplotype, you can have many copies of C4a or many copies of C4b, or one but not the other. And so what Ashwin and Steve did is they actually quantified that and looked across many, many thousands of people and started to ask, how does the form of C4 that you have, does it in any way relate to schizophrenia risk? And the other thing they discovered was that the more copies or the more C4a one has, the more C4 protein or the more C4 gene expression was going on in their brains. And putting all this together and combining this and mapping it back to the genetics, what they discovered, and I'm putting now all of their work into one very simple diagram just to show the main finding, which is those individuals that express multiple copies of C4a make more C4, and they have the higher risk of schizophrenia versus those that don't. So this is really an important discovery because it identified a gene, and it also showed directionality, right? But now the big question, and you're probably wondering this, what the heck's C4? Like, what does this mean in terms of the biology? What does complement do in the brain? Or what is it doing in the brain? And this is really, in a way, where we came in as collaborators because we'd been studying complement in the context of brain development. And it turns out the traditional role of complement in your immune system is if you have an infection or a pathogen, complement are these groups of secreted proteins, and they essentially tag that pathogen, and it can become removed by these circulating macrophages in your immune system. They're like Pac-Man. So that's the traditional way that you actually help fight off infection. But what we now know is that a lot of these complement proteins, including C4, is also expressed in the brain normally. In fact, there's a lot of these complement molecules and other immune molecules you would not be thinking about would be in your brain are there normally, and they have a lot of diverse functions in brain development and synapse function. And so in the brain, what we and others have shown is that these complement proteins, similar mechanism is co-opted in the brain, and the idea that these complement proteins now are tagging these extra synapses for removal by these immune cells, kind of like the Pac-Man in your immune system, but they're called microglia. And then that leads for these microglia to remove these complement tag synapses. And so this is just a real image of what complement looks like in human brain cells or in the human brain, just showing that these complement proteins, you can stain for them, and they decorate synapses and neurons, and that leads to a find me or an eat me signal that then tells the microglia, remove me. So this is one mechanism that we and others have shown plays a role in normal development, based largely on work in animal models. So now the big question is, does any of that biology relate to the genetics and those clinical symptoms and other observations I told you about a moment ago? And this then puts forth a possible hypothesis, although there's much work to do to really be able to test and validate this hypothesis. So the idea is that maybe this overactivation of complement, because of these genetic results, leads to too much of a good thing, too much pruning, and that might in some way relate to some of the schizophrenia pathobiology. And also, intriguingly, could pruning help explain this onset of schizophrenia during development? So there's many, many questions that this work has led us to try to understand and try to unpack. But I think now we're at least in a position, because of the genetics, where now we actually have a foothold, a molecular foothold, to start to try to test and validate some of these hypotheses. And moreover, the challenge now is, how do we validate this? How do we go about that? And this is now work in the next 10 to 15 years that we're thinking a lot about. I gave you one example, the C4 story, but there are many other pathways and genes that you want to start to understand biologically what's going on, and then you want to try to validate that and test this in humans. And one way we might imagine doing that in the context of complement, for example, is since these are secreted molecules, we might be able to read out some of these complement changes in the cerebral spinal fluid, or CSF. So if you've got a spinal tap, a lot of the proteins that might be building up or changing in your spinal fluid are sort of a proxy of what's happening in the brain. So perhaps that's one way we could start to test whether elevated complement or other synaptic-related proteins are elevating during this time. And ultimately, we want to be able to figure out ways to correlate any of these observations with changes in synaptic loss, cortical thinning, and cognitive defects. And that's really where we're at. We're in the process now here at the Stanley Center in an effort led by Steve Hyman, the director of the Stanley Center, and many others, to start a consortium, a biomarker consortium, to try to collect and collaborate to try to gain access to CSF and other tissue so that we can start to test some of these biological hypotheses based on the emerging genetics. And unlike Alzheimer's disease and other diseases, there's been a lack of access to human tissue, to human fluids, in a way that we could start to track this over time. And I think this is one of the reasons why we want to try to mobilize and collaborate to try to build a consortium to try to get us access to those samples. So C4 is just one potential mechanism, one of many genes and pathways that might be involved here. And I told you about that one because we know something about the biology. But now the question is, how do we come back and ask whether some of the other genetic pathways or genetic variants might also start to nominate other biological mechanisms? So could there, for example, be a convergence of risk genes onto a limited number of pathways and a limited number of cells? That's one of the questions we're starting to ask. Because once you have a pathway, and a biological pathway, that's something you might be able to start to think about targeting. And so stepping back, this leads to another big question that I sort of posed at the beginning. Okay, even if we have these pathways, which cell and which circuit? How do we go after that question? Again, stepping back to the 100 billion cells, how might we start to bring together the genetics, the biology, to identify vulnerable cells or vulnerable synapses? And this is where another really important innovative technology that was developed here at the Broad is actually opening up new paths and new directions. So stepping back again, thinking about the human brain, there's over 1,000 different types of cells in the brain. They vary in size. How do we start to identify those vulnerable cells? And it turns out that many people have developed ways to be able to take a brain and break it down into inside of individual cells into sort of what they call a smoothie of all of the cells mixed together, and you can read out gene expression. So what Evan McCosco, Steve McCarroll, Aviv Regev, and others here at the Broad developed was a technology to start to read out gene expression of individual cells from the brain so that you can start to bring that back and understand pathways and mechanisms. So this was a technology called DropSeq that was developed a number of years ago, and now this is the basis of many other similar technologies that are happening all over to try to address this question of reading out gene expression in individual cells. And the idea is that you take a complex tissue like the brain, you can break it up and dissociate it into individual cells, and then they came up with a really clever way to then put those cells into little droplets and then combine those droplets into a microfluidic system that then has barcodes that can read out their gene expression in tens of thousands of cells per experiment in this high throughput way. Now, instead of having the smoothie, you can have the fruit cocktail. This is an analogy that I love that Steve McCarroll and Evan often use, and Aviv as well. But it helps to understand the fact that now you can start to break it down into individual gene expression. So what do you do with this, and what does that data look like? When you then take all of those cells together and all of the different genes they express and put it onto a two-dimensional plot, you get something like this. And what that shows you is that each of those colored clusters or groups represent cells that have similar gene expression patterns and different from the ones of different colors. And that enables scientists to go in and say, okay, well, what's different about all of these different groups? And what happens is that each of those colors represents different cell types and even different cell states. So imagine if a cell of one type suddenly started becoming in a more disease-relevant state. You could actually see changes in gene expression in those individual cells. That's important because that's going to start to give us potentially new markers of these cells, that not only we can start to identify what those markers might be, but we could also then go into the cell, into the brain, and maybe even come up with new biomarkers for those cell states. So lots of questions and lots of ways that this single cell techniques can start to teach us about the mechanisms of psychiatric disease. We can, for example, start to localize some of the genetic hits into specific brain cells and circuits. So we want to know which of those cells express risk gene A. We can then read that out in a plot like that. Second question is we can ask, how do these different disease states correlate with different stages of dysfunctional states in the brain? So we can start to perturb and look in patients that have the risk gene versus not and see how the circuitry in the cell states change. So that could give us an unbiased look at disease mechanism. That could lead to the discovery of new biomarkers that we could read out in numerous ways. And it's another way of also evaluating disease models. And I'm going to end now by just saying that what we really now need to do is start to understand and model disease mechanisms. And for the last many years, the field largely has used animal models like mice, which offer a lot of advantages because they're amenable to genetic manipulation, and you can track and dissect mechanism in an animal model like a mouse. But the problem is, if you really want to try to model complex polygenic disorders like schizophrenia or others, the mouse just isn't going to work, in part because the genetic background makes a huge impact on the results, right? And so generally, we go into these mouse models where you know a gene and you knock it down and you ask what happens. But in the context of disorders like schizophrenia, where there's many, many genes that contribute together, the genetic background is really critical. And so that has led to this question of, can we come up with better models to model disease mechanism in human? And since you can't go in and do this non-invasively in the human, one of the things that's really been transformative is the ability to use stem cell or human cell-based models, in vitro models, to start to do this. And so this is technology many of you may have heard of, but one can, for example, take skin cells or blood cells, and you can differentiate them in a dish into these cells of interest, whether that be a neuron or a different cell type, using a different sort of combination of signals. And although many things can be done just by putting them in a dish like this, you can compare, for example, patients versus control cells, or you can manipulate a particular risk gene and ask how that changes the function of those cells. But what's now been able to be quite transformative to the field is this new way of actually generating not just individual cells, but putting them in these bioreactors to make these 2D mini brains or brain organoids that enable you to start to model not only the individual cells, but how they interact, how they form synapses, how they communicate with one another, and how they develop. And this has really been, and is in the field, I think, going to be a new way to start to take those genetic leads to try to understand the underlying genetic and biological mechanisms. So this is just an example of work from Paula Alada's lab, who's here at the Broad Institute, and a lot of colleagues, just to show an organoid. After several weeks, you can start to see different cell types and diversity. In fact, when they sequenced, again using single-cell sequencing, it turns out that almost all of the cells in the human brain are represented in these organoids. And they have structure, they have organization, they make connections. And there's actually, Paula's recently, her lab has developed ways to really make these quite reproducible. So you can start to imagine all kinds of ways to start to use these models to start to interrogate disease mechanism. For example, you could imagine getting skin cells from patient or control or individuals with high versus low polygenic risk, and then comparing the organoids from those two. And then you can do the similar example where you could actually, sorry, went back one. Let me see, there we go. We could also imagine manipulating, using genetic engineering, some of those risk genes and asking how that influences cells and circuits. And so this now enables you, using a lot of the technologies I just mentioned, to start to interrogate more deeply mechanisms, including connectivity and how synapses are developing and how synapses function, which brings us back to where we all began. So there's much to be done, but I wanted to just illustrate at least a couple of the innovations and new approaches that members of the Broad and over the last many years have developed that are now enabling I think really a lot of progress in trying to link the biology to the genetics. And of course, the ultimate goal of all this is to try to translate biological insights into new biomarkers and therapies. And although it may seem a bit far away, although we still have quite a lot of work to do to understand the biology, I think we're really well poised because of these approaches to start to really dig deeper to identify new potential disease mechanisms that could then lead to targets or new predictive biomarkers. And I think this is really going to take a collaborative effort across many different labs and groups, and also work here at the Broad with different platforms and working with the therapeutics department in platforms is going to enable us to start to work together to move this work forward. So I just want to end by thanking all of the many of thousands of patients and families who have volunteered for and contributed to these studies. And I want to also end with a very special thank you to Ted and Veda Stanley, who are really amazing. We're amazing philanthropists and who made several really large and generous contributions that enabled the Stanley Center to be founded in 2007. In fact, their second contribution was of $650 million, which is the largest ever for neuropsychiatric research. And that has led to the Stanley Center and a lot of the work that I told you about today, which is really aimed at trying to understand more deeply research that's going to help move forward the development and the identification of new therapies and biomarkers. So I want to end there, and I'm happy to take any questions. Thanks so much.
Thank you, Beth, and thank you, Ben. Come on up on stage,
Sure,
and the floor is open. If you have a question and you're in the audience, please either raise your hand for a mic runner or text your question to our question phone number. And if you're on YouTube, also, please drop your questions into YouTube chat.
Two very nice presentations,
right away.
thank you. I'm assuming that a lot of the mutations you're talking about are germline mutations that have been inherited. Has there been any work on somatic mutations to track whether genetic mutations occur as the disease progresses?
Sure. Excellent question about somatic mutations, mutations that are arising as risk factors for schizophrenia and other mental illnesses. So in autism, mutations in a gene called PTEN lead to proliferative advantage in neurons that can then give rise to a kind of developmental delay kind of presentation in a more somatic rather than germline sort of landscape. It is worth remembering that the somatic mutations are unlikely to be indexed by the familiality, so the kind of twins and family members and those sorts of things. Unless there's a somatic mutation that occurs in the parent that is then present in multiple germ cells that then gets passed on to the offspring, it will more typically focus on, that'll more typically reflect genetic variation that is inherited more widely. The hardest thing about interrogating somatic mutation in particular is access to brain tissue. So even if there was you would have to have very widespread mosaicism to be able to see a signature as a risk factor until you do it, in kind of an autopsy context. And so that's really limited our ability to get a good view on the role of that class of variation in risk for illness.
All right. Question over here. Yep. Oh, I was pointing to the gentleman a couple of rows up, but you're next, sir.
What about the role of epigenetics and non-genetic influences? Particularly unfortunately,
Sure.
Like trauma being passed down, that sort of thing.
Yeah.
We got to go into the middle.
Yeah. Into the light.
Yeah, there you go. So for epigenetic factors, again, quantifying epigenetic factors at scale is very challenging. Some of the genetic variants that we're identifying as risk factors are likely to act through epigenetic mechanisms. So they're things like chromatin remodelers that are going to reshape the way that the DNA is structured, which will have then potentially some impact on what genes are expressed and when they're expressed, although we don't have super clear evidence of exactly what each of those steps looks like. In terms of other indications of epigenetics, if we look at parts of the genome that are, sort of unraveled in neurons, that's where we tend to concentrate heritability. So that is to say that the sort of chromatin remodeling that we can see in terms of what parts of the genome are active does seem to point a little bit more to genetic variation that's influencing risk for things like schizophrenia or other forms of mental illness. But in terms of being able to measure epigenetics directly, again, we're stymied by our inability to access tissue and the comparative cost of doing those kinds of characterizations compared to the collection of genetic information. I think the expectation is that it will play a role, and it certainly plays part of that role in how you go from genetic variant to actual biological mechanism that's driving risk for disease. But I'd say that's more of a black box at this point.
Yeah,
Beth?
great. I just wanted to add to that, in the same way that technology is going to enable maybe us to get a little bit closer to answering that question, there's now going to be ways with dissociating cells and looking at the single cell level to be able to relate those single cell changes in transcriptomics or gene expression with the epigenetics. And I think those two together might also point to new pathways and new mechanisms. And then the other thing just to say, which is the non-genetic ways that some of these pathways could be influenced. What we didn't cover today, just because it's a bit of a black box, but the idea that environment can influence the brain and non-genetic sort of perturbations or challenges might also, at the right time, for example, in development, could in some ways push the scale. And so one of the questions I think we're well poised to ask now is, as some of these pathways now are coming forth, we can imagine, how do things change when you do and don't challenge the brain or the environment? How does that change the outcome, the functional consequences? And I think that's one of the questions we'll be particularly interested in doing with respect to developmental mechanisms, for example.
Thank you for very interesting talks. Because of the importance and effects of thinning the neurons, as you mentioned, I was wondering whether the artificially grown brain organoids and or animal models can help because of the many trials you could probably execute on them. That's my first part of the question. And the second part is, how much is known, if any, about influences that increase or decrease the rate of thinning, and by area or just overall?
Yeah,
and is it by area or overall? Can
sure.
you focus?
Yeah, so we don't know much. I'll just take your second one first. Based on the brain imaging studies, we certainly know there's this developmental trajectory, as I showed in that image that showed, in a way, those changes in maturation was actually represented thinning that happens over development. But because not everybody, especially at that what they call prodromal stage, that data is often just not available to know when that happens, to be able to make and increase the sample size that would start to give us more power. So that's an important point, is not knowing, how prevalent that is, and also are there certain brain regions and circuits that are more vulnerable to thinning than others? So I think that's a really important, still open question in my mind. But going back to your first question, can we use some of these human cell-based models to try to model things like thinning and pruning? I think we have the capacity now, especially with the generation or the next generation of these organoids, which the Arlotta lab, for example, has shown actually do form connections, and you can see these changes happening over development. If you were then to imagine perturbing some of the risk pathways, you could ask questions like, does this change the amount of synapse loss or the way that the synapses are communicating with one another? This just wasn't possible until you developed ways to make these networks actually form from human cells. So I do think the human organoids may be a way forward to try to understand those circuits. And that's really, I think, the next step in taking those organoids to the next level, to understand the functional consequences of perturbing these pathways.
Thank you.
Yeah.
I'd like to jump in with a question from the texting line.
Okay.
Beth, somebody texted in asking, given the relationship with C4 and schizophrenia, would you expect or do you see any correlations with autoimmune diseases?
Yeah, so I cannot address that or answer that directly. However,
I can.
I think that Ben can on the genetic side, in that there is certainly the lupus side of the story, and so maybe you're going to mention that as well.
Yeah, so I think, the first thing to put on the table is that most of the immune disorders associate with what we call classical alleles of HLA. And these classical alleles go back to before we had like genotyping or sequencing technologies. People figured out that there were different kinds of HLA alleles, and they could do tests for those alleles. And these alleles are governing basically, they're things that your cells are presenting to identify itself as itself. And a lot of autoimmune disease risk is trapped in that part of the genome. And that kind of classical allele in the HLA or the MHC region was not what was associated to schizophrenia. It's actually a really different kind of immune gene than the more typical association in the HLA. That being said, Beth quite correctly highlights this emerging work on lupus that Steve McCarroll and Nolan Kamataki and others in his group have been pushing on, looking at the role of C4 as it relates to risk for lupus. But by and large, most of the autoimmune disease risk, at least in the HLA, is coming from the so-called classical alleles rather than the C4 copy number, which is a different genetic variant in that same region.
Thank you. All right, any more questions in the room? I see lots of hands.
Go over to the right side.
Hi. My question was related whether we can see what cells or synapses that are destined to being pruned away expressed on the cell membrane, but you already answered, I guess. So.
Yeah, but I'll just add a little bit to that point, because that question you just asked is one that we're really interested in understanding, because the question of what's bringing these complement to synapses is still an open question. Trying to understand what the receptors are, the binding proteins, could then lead to ways to then target that particular aspect in synapse function or synapse loss. So we've been doing a lot of, for example, biochemistry and mass spec, pulling down synapses and trying to figure out what's coming down with the complement proteins to try to identify new potential candidates. So stay tuned, but I think that's going to be an important question.
Down here, Corey. Actually, I'll just run over. Here you go.
You mentioned that this work is a long way from clinical application, and I appreciate that. But is there anything I can tell the families that I teach who have loved ones with serious mental health conditions about length of time, sort of 10 years, 50 years? Can you give any more detail to that? Thank you.
Do you want to take that one? That's a tough one.
Thanks, Beth.
Ben's like, great.
Forecasting is notoriously difficult in the drug development industry and universe. There are a lot of reasons why things go wrong, and I don't want to be in the position of giving false hope. I think that, a therapy in 5 years is very, very unlikely, maybe a little bit more likely in 10, and we have a lot of people working really hard to make it a reality in 15 to 20. But there are no guarantees in this, and we know a lot less than we need to know to be able to really get there in a kind of direct path. There's still a lot of unknowns between where we are now and how we might think about developing therapeutic intervention. And that's why I would counsel caution. If they're looking for the moral support, though, there is progress being made. We know the genetics is working. When genetics has worked in other parts of human health, that has translated to, in a time lag way, serious improvements in therapy. Cancer is probably the most notable example of precisely that, where understanding a lot more about what the genetic landscape and genetic risks were has really transformed the way we think about and treat cancer. And we're going through that same transformation right now in mental illness. Yeah. And so that's the hope I would hang on, rather than this time in 15 years' time will be guaranteed.
Yeah, I wouldn't mind just adding another optimistic note to that, in that I think one of the things that's been really needed to move therapeutics forward is biomarkers. And if we could identify some biomarkers, whether that be fluid, blood, or even imaging biomarkers that could help us stratify patients to identify those at risk, that could then lead to ways be able to target those populations and move things forward when the time is right. And there may also be converging pathways or mechanisms that are coming from other diseases. We can even imagine, if we were to identify a mechanism in the right people, that you could imagine repurposing. As long as you have those biomarkers, that's the key. That, I think, we're making progress on, because we're starting to really pull together, and the field is in a position where we can start to at least collect samples and start to build the cohorts. And I think that is something quite optimistic and positive.
I think we have time for one more question from the room. This gentleman right over here.
Thank you. So based on this new data that you have, how likely do you think that these complex diseases are in fact a set of many different diseases that have different causes, but that in the end have the same, look the same, or have the same phenotypes in a way? Thank you.
I'll take that.
Okay.
Sure. So the question is, this is a complex disease that we're looking at, or complex diseases. What are the chances that it's not one thing, but basically lots of different sub-things that are all sort of mixed up together, I think is the essence of the question. And that's an excellent question. And it's very difficult to know very clearly that it's one thing or multiple things. I think that what we can say is that the evidence of heritability, both from the twin and family studies and from the sort of genome-wide association studies, gives us some confidence that there's at least some coherence. Certainly when we're taking tens of thousands of patients with schizophrenia and comparing them against tens of thousands of controls, we do see some patterns of difference across the multiple different genetic loci. Now, that's not the same as everyone has exactly the same flavor of risk profile. They actually all have individual, unique risk profiles. But you might be able to think of it a little bit more like something like, say, a heart attack, where you might get a heart attack because you have really high cholesterol. That might be your sort of primary driving risk factor. Or you might get a heart attack because you smoke a bunch. And both of those are clear risk factors. But when it comes to having the heart attack, if you smoked a bunch and ended up having a heart attack versus had really high cholesterol and had a really bad diet and had a heart attack, it still presents the same as this sort of heart attack. Now, the root causes are slightly different, but the downstream sort of disorder or disease is quite similar. And that might be one way of thinking about the kind of polygenic risk landscape. It's not one unitary construct. There are probably lots of different biological processes and mechanisms, things like synaptic pruning or other synaptic strength maintenance that isn't being governed by pruning but may relate to risk for disease. All of those different biological aspects might give rise to the same presentation, but an individual patient might have a little bit more of one versus another, and that might be where we end up in the landscape for schizophrenia.
All right, well, thank you, Beth, thank you, Ben, and thank you all of you for coming tonight. And we hope you'll come back for our next talk on November 14th on the development of therapeutics. Thank you very much.